Thank you for joining us today. This is Brian Hall, Product Manager of the Imaging Flow Cytometry Instruments at Luminex Corporation, and I'll be joined by Katie Gillis, Product Manager of the Guava Muse Systems, and Stephanie Brunelle, Product Manager of the CellStream, and together we will be reviewing imaging and flow cytometry applications critical to the field of immuno-oncology. The Amnis imaging flow cytometers are high-throughput microscopes that collect bright field, dark field, and up to 10 channels of fluorescent imagery at a rate of 5,000 objects per second. This generates a large image database that we can then use statistical image analysis software to go in and effectively mine that database for unique morphologies based not only on fluorescence intensity, but the location of that fluorescence as well. And this enables some novel applications that you can do with this type of technology, including cell signaling applications, colocalization, apoptosis, and many others. This combination of flow cytometry and imaging allows researchers to gain a deeper understanding of the subcellular processes within their samples. It allows for a limitless number of applications to gain a deeper understanding. It allows researchers to make novel discoveries, learn the mechanism of actions, and effectively get the science right. In this first example, we're going to look at T-cell activation after binding to antigen-presenting cells and demonstrate NF-kappa B translocation to the nucleus. These T-cells are harvested from the thymus of transgenic mice that are sensitive to ovapeptide, where treatment with ova enables the T-cell to bind the antigen-presenting cell. For the ImageStream experiment, we're going to label the T-cells with CD8, the antigen-presenting cell with CD45. We're going to label the nucleus and NF-kappa B as well. And then in the analysis, we're going to be measuring the percentage of CD8 positive T cells actively involved in conjugate formation with an APC and showing activation indicated by translocation of NF-kappa B to the nucleus. Now the first part of this experiment begins identifying the doublets versus the single cell population. So the population in the upper left of this plot shows that these are the single cells, and the R3 population are the doublets. Now these doublets could be coincident conjugates or T cells actively engaged with an antigen-presenting cell. So to verify that we have a T cell APC conjugate, we're going to look at a CD45 versus CD8 intensity plot and identify true conjugates. We can verify that population in R5 by looking at the imagery showing one T cell and one antigen-presenting cell. Now the next step of this analysis is to identify the nucleus of the T cell. To do this, we can use a mask, and a mask simply defines a region of interest. Ideas has a number of different masks that you can use to look at different subcellular compartments. And in this example, we're going to be looking at a combined mask. For this analysis, we're going to be masking the CD45 APC and the nucleus of both the T cell and the antigen-presenting cell. We can then overlay the CD45 mask on the nuclear mask and remove it from consideration in the analysis. So we've effectively masked the nucleus of just the T cell and not the antigen-presenting cell. We then use similarity, which is effectively a measure of nuclear localization of NF-kappa B, on these true conjugates. And by looking at similarity of this new mask that we've created, we can see that events that have a low similarity score show that most of the NF-kappa B is sequestered to the cytoplasm, whereas after treatment with a high dose of ovapeptide, we get an increase in our similarity or translocation score, and simultaneously the imagery demonstrates that NF-kappa B has migrated into the nuclear compartment in just the T cell, showing T cell activation. We can then use the similarity feature over time in different doses to create a dose curve and measure the EC50 after treatment with different OVA concentrations. In this next experiment, we'll be looking at how morphology can be used to measure the cell cycle progression, the apoptotic index, both before and after treatment with a chemotherapeutic agent. Now this is a cell line, U2OS cells, that have been genetically modified using zinc finger nucleases to add fluorescent proteins to both the lamin B1 protein on chromosome 5, the tubulin A1B protein on chromosome 12, and the actin B protein on chromosome 7. The U2OS cells are effectively an adherent cell line. So to maintain the adherent morphology, these cells were fixed on the plate and manually brought into solution. The cells were also stained with DRAC5 to label the nucleus so that we could look at chromosome formation and progression throughout the cell cycle. Each phase of the cell cycle can be characterized by total DNA content and the morphology of the actin and tubulin fluorescence coming from the fluorescent proteins in these cells. And as you can see from the imagery, as the cells are in G0, G1, they maintain the adhered morphology. During S phase, they gradually begin to increase the amount of DNA in their nuclei. At G2, they begin to prepare for mitosis, and then in the prophase, you can see the cells begin to round up off the plate and start to form chromosomes. During metaphase, those chromosomes align on the metaphasal plate, and in anaphase, you start to see separation of the chromosomal centers. And in telophase, you can see in the bright field imagery that the two cells are beginning to pinch off and form daughter cells, and then returning back to the G0, G1 phase, you can see that the cells then lay back down onto the plate. And so by capturing an image of each of these cells in solution, we can get a really good indication of cell cycle progression throughout each phase of the cell cycle. Now, if you look at the morphology of these cells during mitosis, you can see that the nuclear envelope dissolves as the cells begin to progress through mitosis. You can see that tubulin starts to polymerize, forming mitotic spindles to bind to the chromosomal centers. And you can see that the cytoskeletal structure breaks down. Now, if you look at a total intensity plot that measures the amount of fluorescence coming from each of these monomers, you can see that the monomeric form is still retained in the cytoplasm. So just looking at total intensity alone, as in figure A, is not enough to characterize the phase of mitosis that these cells are in. That's because the monomeric form is still in the cytoplasm and fluorescing. So the total amount of fluorescence is the same. What's really changed is the morphology of that fluorescence. So in the plots along the bottom, you can see we can utilize morphometric features to, in the case of plot B, look at dissolution of the nuclear envelope. C, you can see there's a dramatic increase of this tubulin formation morphology. And in plot D, you can see that the cytoskeletal structure dissolves, breaking down and allowing the cell to progress through the phases of mitosis. Vinblastine is a chemotherapeutic agent used to treat Hodgkin's lymphoma, and it functions by binding tubulin to prevent proper formation of the mitotic spindle and block cell cycle progression. It also induces apoptosis. So in this example, we can look at the morphological effects of treatment of vinblastine over time. So here we treated the U2OS cells, were treated for up to 24 hours with 0.5 micromolar vinblastine. The apoptotic index is measured by looking at nuclear condensation and nuclear fragmentation as indicated by the imagery. In the lower right-hand corner, you can see using the nuclear morphology, we can calculate an apoptotic index, where at 0 hours, we have a background of about 3% of the cells are apoptotic, and after 12 hours of treatment with vinblastine, about 21% of the cells are apoptotic. So we can use this plot to create our apoptotic index and measure that over time to really quantify the effects of vinblastine on these subcellular components as measured by their morphology. Acute lymphoblastic leukemia is characterized by an overabundance of immature B cells. In this experiment, we'll be looking at T cells modified with a chimeric antigen receptor that can target these CD19 positive B cells and induce cytotoxic effects. So here, CRISPR-Cas9 was used to enhance cultured lymphocytes to target the CD19 B cells. CAR-T cells were also incubated with and without ICAM, which can be used as a blocking agent. And for the ImageStream experiment, we'll fix the cells. We'll label the T cells with either CD8 or CD4 and the B cells with CD19 and compare which of the T cells, either the CD8s, the CD4s, or the CD8-4 combination, does the best job of identifying the target B cell. We can then quantify conjugate formation and the cytotoxic effects, as well as look at ICAM blocking. To analyze this experiment, we want to begin by characterizing the T cell subsets in this sample. So thymocytes will express either CD4 single positive T cells, CD8 positive T cells, or early thymocytes can express CD4 and CD8 simultaneously. After gating on these three different CAR-T subsets, we can compare their ability to bind to B cells and induce cytotoxic effects. So in the middle series of dot plots, you can see approximately 18% of the CD4 CD8s are binding to the B cell. Looking at the corresponding imagery in the top panel, you can see these cells are double positive for CD4 and 8, and they're attached to the B cell. About 14% of the CD4 positive CAR-T cells are binding to the B cell, and about 7% of the CD8 positive CAR-T cells are binding to the B cell. So after compiling the data from that analysis, we can now start to compare the effects of treatment with ICAM and look at that over each subset of CAR-T cell. So if you look at the CD4 CD8 CAR-T cell, that seems to show the greatest affinity for the target B cell. If you look at the CD8 only positive CAR-T cell, it seems to have a relatively low affinity for the B cell. And clearly, ICAM inhibits CAR-T binding in each of the T cell subsets. So utilizing the image cytometry and the imagery, we can not only characterize CAR-T binding its target cell, we can also look at cytotoxic effects in downstream experiments, and we can use the imagery to really characterize the effectiveness of our CAR-T cells when treatment with cancers such as ALL. So that was the last example that I had showing the benefits of imaging flow cytometry when analyzing your immuno-oncology experiments. Next, I'll turn it over to Katie Gillis, who has some great applications that she describes using the Guava Muse systems.
Thank you. The Guava Muse and Guava EasySite systems have many applications for the immuno-oncology market. We are now going to discuss some of those applications. The Guava Muse and EasySite instruments use a patented microcapillary system that provides a simple and reliable design. The microcapillary eliminates the need for sheath fluid in our flow cytometers. Utilizing our patented fiberglass flow cell, the laser light hits the flow cell. It allows for scanning of every particle that flows through the system. We then combine this with a positive displacement syringe pump, allowing us to determine absolute counts because we know exactly how much fluid flows through the system every acquisition. In addition to the accurate cellular counting the microcapillary and syringe pump design provides, the technology allows for small sample sizes, which preserve your precious cellular samples and produce low biohazardous waste. The Muse and EasySite technologies provide pre-validated kits that allow for the identification and enumeration of both whole blood and PBMCs. These assays are mix and read and take as little as 10 microliters of sample. The sample is mixed with one of our pre-formulated kits and acquired on our Muse or EasyCyte instruments. Each instrument has a predefined acquisition and analysis template that allows the user for simplified acquisition and analysis. The Guava counting assays, highlighted here, utilize our proprietary mix of dyes to exclude cellular debris and identify the count of nucleated as well as debris. We also identify viable, dead, and dying cells. Unlike assays that utilize trypan blue, the Guava counting assay uses a DNA intercalating dye that indicates any membrane disruption and can be an early indication of cellular distress. The Guava Immunology assays are developed to provide the user with CD4, CD8, or B cell concentrations and the percentages in the overall lymphocyte population. These are also mix and read assays that can be combined to determine all of the various populations. Here we show data for the Muse count and viability assay on PBMC samples. A simplified output for the assay is shown in the upper left. You can see here that the assay provides viable cells per mil, percent viability, total cells per mil, as well as dot plots for clear separation of the various populations. The graph shown here compares the cell concentration on the Muse cell analyzer versus a predicate method for both fresh and frozen PBMCs. As the data shows, similar performance from the Muse count and viability assay was seen when compared to the predicate method, with both R² and slope values over 0.9. Additionally, the table in the lower left showcases the percent CV for replicate measurements. The percent CV shown for both fresh and frozen PBMCs are well below 10%. This data indicates highly precise and accurate measurements that can be attained while using the Muse cell analyzer for count and viability measurements. Results for the Muse immunology assays are shown here. The Muse immunology assays provide absolute counts, total lymphocyte counts, and population percentages based on the total lymphocyte population for either CD4, CD8, or B cells. Two of the assays are highlighted here. The dot plots in the upper left highlight the results for either the CD4 assay or the CD8 assay. The lymphocyte population is shown on the y-axis for both plots, and either the CD4 or the CD8 population is shown in the x-axis. Very clear separation for the lymphocyte populations is shown for both assays, as well as clear separation for the CD4 as well as the CD8 populations. A comparison of the CD4 counts for a predicate method versus the Muse cell analyzer is shown in the graph on the right. You can see that based on the linear response that is shown, based on both the slope as well as the R² value, that comparable results are seen on the Muse cell analyzer. Lastly, we evaluated the CV results for the count, the percent lymphocyte, as well as the count for the total population. These results were compared for 3 operators across 3 instruments running 4 samples in triplicate. You can see that the CVs for all of these populations were well below 5%, highlighting that not only the precision within the assay, but across users and instruments is extremely high. We took immune cell identification and enumeration one step further on the EasyCyte platforms, which have the ability to evaluate up to 12 fluorescent channels at one time. To do this, we did a T, B, and NK analysis using a blood control from an external vendor. The dot plots show the gating strategy and separation of the populations for either the CD check plus sample or the CD check low sample. You can see that CD45 positive cells were gated into a CD3 plot where we looked at CD3 negative and CD3 positive cells. Our CD3 positive cells were then gated into a CD4 versus CD8 plot, and our CD3 negative population was gated into a plot where we looked at CD19 B cells and CD16 and 56 positive NK cells. You can see that the separation for all of the populations are very clearly separated for both the plus as well as the CD check low sample. The graphs on the right show the counts for the various populations when compared to the vendor specified values. The vendor lower limit is shown in red, the high limit is shown in pink, and the average value is shown in orange. The easy site results are shown in blue. As these results show for both the CD check plus, low, as well as regular samples, the counts are well within the vendor limits. Using fresh whole blood donors, we then decided to not only do a TB and NK analysis on our samples, but also to evaluate the naive and memory populations. Shown here is an example from one such donor. The data shows a similar gating strategy as we saw on a previous slide, gating our CD45 positive cells into a CD3 gate where we looked at the positive and the negative samples, and then taking our CD4 positive cells and our CD8 positive cells and doing a naive and memory analysis on those. You can see that there's clear identification of multiple important immune cell populations from this simple analysis utilizing only 10 microliters of blood. One final area I would like to show today is related to mechanism of action studies during drug discovery, development, and deployment. Here we show an example of the mitodamage assay that combines 3 cellular health markers to assess changes in mitochondrial membrane potential, apoptosis, or cellular death. This example deploys all 3 cell health markers simultaneously. Mitosense red detects changes in mitochondrial membrane potential and is shown as a downward shift in fluorescence, indicating a loss of the membrane potential. Apoptosis is detected using an exon 5, which indicates phosphatidylserine exposure. Lastly, cellular death is detected using 7-AD staining, where there's a loss of membrane integrity. The dot plots shown here show example stainings for the assay on jerkats. Clear separation for each population is seen. The 2 bar graphs show dose responses for either etoposide, a known topoisomerase inhibitor, or styrosporine, a known protein kinase inhibitor, and the corresponding population percentages, with depolarized populations shown in red, apoptotic populations shown in yellow, and cellular death populations shown in blue. The bar graph is shown for both compound depolarization and apoptosis. See similar percentages, but little cellular death was seen. Additionally, at this time point, styrosporine shows a much larger shift in depolarization and apoptosis when compared to etoposide. Therefore, in addition to counting and immune cell subpopulation identification, the Guava platforms can be used to evaluate and assess drug mechanism of actions and effects. Thank you. Now I'll turn it over to Stephanie.
Next, we'll talk about the Amnis CellStream, which is based off of the same TDI CCD technology as the ImageStream and FlowSight, which Brian talked about earlier. The CellStream system is a benchtop camera-based flow cytometer built on the Amnis technology similar to the ImageStream. An image gallery is used to visualize objects in flow. The CellStream is a high-throughput, compact system with a small footprint, and it allows for modular and flexible upgrades. The high sensitivity of the flow cytometer allows for traditional flow assays as well as small particle experimentation. The CellStream can be used for traditional assays such as immunophenotyping. In this particular study, we performed a 16-color immunophenotyping of major lymphocytes and myeloid subsets in human PBMCs and whole blood. As you can see, we easily resolve all expected populations, as well as populations identified by dim markers such as T-cell subsets. The T-cells were characterized into distinct subsets including naive, effector, effector memory, central memory, activated, and regulatory T-cells. The CellStream can be used for traditional assays such as immunophenotyping. In this study, we performed a 16-color immunophenotyping of major lymphocytes and myeloid subsets in human PBMCs and whole blood. As you can see, we can easily resolve all expected populations, as well as populations identified by dim markers such as T-cell subsets. The T-cells were characterized into distinct subsets including naive, effector, effector memory, central memory, activated, and regulatory T-cells. In this study, we used the CellStream to monitor the phosphorylation of signal transducer and activator of transcription, or STAT proteins, which are transcription factors that migrate to the nucleus upon phosphorylation and alter gene expression. STAT proteins are known to be stimulated by certain cytokines, such as interleukins. STATs 3, 5, and 6, which have been associated with prognosis in cancer and other malignancies, were simultaneously monitored in CD4 positive T cells in these experiments. Whole blood was stimulated with interleukins 2, 4, and 6. Red blood cells were lysed and the remaining leukocytes were analyzed with antibodies against phosphorylated STAT proteins, specifically phosphorylated STAT 3, 5, and 6. The gating strategy is shown here for identifying CD4 positive cells. The phosphoflow results show that IL-4 stimulated STAT6, as in the top panel, and you can see a shift in fluorescence outlined by the box. IL-2 stimulated STAT5, as seen in the second row, and IL-6 stimulated STAT3 in the third row. On the bottom part of this slide, you can see a mix of interleukins phosphorylated all the STATs we investigated. The unstimulated blood is shown in gray, and this is the negative control. Next, we'll shift gears and talk about extracellular vesicles, or EVs. EVs are membrane-derived structures that include exosomes, microvesicles, and apoptotic bodies. Exosomes range from about 30 nanometers to 100 nanometers. Exosomes have been shown to transfer molecules between cells and have the potential to transfer signals between cells. Exosomes are released under normal physiological conditions. However, exosomes are also believed to serve as mediators in the pathogenesis of neurological, vascular, hematological, and autoimmune diseases, as well as cancer. Quantifying and characterizing EVs is challenging due to their small size. High magnification microscopy such as EM can be used to analyze EVs, however, it's very low throughput. Additionally, PMT-based flow cytometers have a difficult time detecting EVs since they are not highly sensitive. Imaging flow cytometers that use a charge-coupled device or CCD camera can detect EVs, such as the CellStream. In order to immunophenotype EVs, we follow this workflow using red blood cells, or RBCs, and platelet-derived EVs. The EVs are obtained from Solarcus Biosciences. The RBCs and platelets were washed and treated with a calcium ionophore to induce vesiculation. The vesicles were then isolated via centrifugation. The resulting EVs were labeled with either an anti-CD235 antibody with PE or a CD41 antibody with APC. The labeled EV samples were serial diluted as shown here. Samples were then acquired on the CellStream for 3 minutes per sample with 100% laser power. All proper controls were run including antibody only, buffer only, and detergent controls. These controls were run exactly as the EV samples. Samples were run in duplicate on 3 CellStream instruments. Shown here in the top row is the gating strategy for identifying the potential EV populations for both the RBC and platelet-derived EVs. The population ranges from about 50 to 200 nanometers with a peak around 100 or 120 nanometers, with the platelet EVs having a larger tail. The bottom row shows the serial dilution data. The 1 to 60 dilution on the far left has a significant amount of coincident events, so that concentration is too high. EV concentration was measured for each of the dilution controls. These dilution controls were used to check for swarming. Swarming occurs when multiple EVs are detected at once or as a swarm, and not as single objects, which is how we want to detect EVs. As you can see here, both sets of dilutions show a linear decrease in the potential EV population using the CellStream for both the PE and the APC labeled EVs. Additionally, the EV mean intensity was measured for the dilution controls. For both PE and APC, the EV mean intensity remained consistent for each dilution. The dilution controls shown here indicate that the potential EV population consists of single objects. In EV research, controls and dilutions are just as important as your samples, since these help determine what is artifact or background noise and what is a true EV. Shown here are the data for the PE-positive EV samples and controls and the corresponding serial dilutions for all. As you can see, the average of objects per microliter for the EV samples are significantly different from all of the controls for all dilutions. The low concentration measured for these background controls validates the EV population. Similar to the PE data, the APC-positive EVs and corresponding controls show a significant difference, and therefore the EV population is validated here as well. Since the CellStream has such high sensitivity and can easily detect EVs, it is a great system for investigating EV cargo as well. The data shown here are from a study investigating EVs derived from human embryonic kidney cells. These EVs were stained with the exoglow protein and exoglow RNA labeling reagents to detect protein and RNA cargo on the EVs. On the left part of this slide, the plots and histograms show the potential EV population positive for protein cargo, colored in green, and RNA cargo, colored in orange. On the top right, dilution series data show that for both protein and RNA positive EVs, there is a linear decrease in signal with extremely low background, validating the labeled EV population. On the bottom right, we see stable mean fluorescence of the positive events for EVs with cargo in the serial dilutions. Protein cargo is shown in green and RNA cargo is shown in orange. The same laser intensities were used for all samples. This indicates the measurement of individual EVs and not swarms of EVs. As you heard from myself, Katie, and Brian, Luminex offers a wide range of flow cytometry products that meet most research needs. As you can see in the table here, we offer a variety of instruments ranging from our cell analyzer to our imaging platforms. Please contact us with any questions about our versatile flow cytometers. Thank you for listening to our presentation.